We're live.
We're live.
Good morning, ladies and gentlemen, and thank you for coming. I got to tell you, we've been doing some planning for this. Our weekend has looked a little bit like this, right? And so meet yesterday, my new granddaughter, Calissy, born yesterday into these crazy times. So with that said, we're really happy you're here with us. We're not really sure exactly, except for this light bar here. We're not really sure exactly what's happening and how long this is going to be. But there's one thing that I think Mrs. Smith and I are in total agreement on, and that is we don't want to be teaching biology in July and August, right? So there's no point in waiting. We're self-quarantining, we're at home washing our hands. You're not supposed to be running around. We figured let's just continue this. Let's keep it going. Let's talk bio. Let's move forward, and that's why we're doing this. So we're really happy that, like, I'm looking right now, there's 43 of you watching, which is great. We'll take it. The rest of the people can watch it on their own time, so you can come in and watch these live streams at a later time. But in hope for some sort of normalcy in our life, we would love for you to join us when the live streams are happening. You can ask questions over in the comments. We'll try and keep eyes on the comments if you have questions. There's other things that we're doing for you. There's office hours. I have my office hours posted in Moodle. I know Mrs. Smith will probably be sending hers out through Blend. If you want to come and talk to us, if you have questions, if you need help with stuff, do that. I told my students on Friday that we expect 40 minutes out of you a day when you're at the high school. And so we are not planning anything that would be more than 40 minutes a day. And if you put the Snapchat away for 30 seconds, you probably can get the stuff that we're going to ask you to do each day done in less time than that. So with that said, Roby1 Kenobi has a question. If you ask the question, we'll get to it. We'll certainly work through that and address any questions we have at the end too. So with that said, I think I covered it all.
Yeah, I think so.
I need to share a little PowerPoint here. I have that posted in Moodle for you too. I know what I needed to tell you. Please, one thing that you might want to do is in Moodle, there's a link to the DNA structure notes. So if you haven't done the DNA structure notes, they're there. Make sure you pay attention to how to label them. That's going to be really, really important why we're in this virtual world for us to be able to find your work. I'm not spending hours searching for it. Neither is Mrs. Smith.
Yep.
Be careful with labeling or titling it so you can find it. But you could probably fill those notes out while we go, so those notes are done. But if you'd rather just sit and listen and fill the notes out when we're done, you can. You can always replay the video. So, Mrs. Smith, I'll turn it over to you to start this wonderful idea of DNA structure, and you just tell me when you want me to click, and I'll click through it for you.
Okay. So this morning we're talking about DNA structure, and I know a lot of my students have gone and done our Ed puzzle already, and there's just some things we have to fill in. But just so that you don't know, when we talk about DNA, a lot of times it's called the blueprint of life. If you think about architects having a blueprint of basically what the structure of the house is going to look like or the building, DNA is very similar. It's basically going to be our structure of what's going to happen and what we're going to look like and do, etc. So when we talk about DNA, DNA stands for, slide suitor, deoxyribonucleic acid. So I kind of highlighted the DNA. And so when we talk about deoxyribonucleic acid, please remember we've talked about this already. We're talking about nucleic acids. This is one of the macromolecules. And so as we move forward, we're now going to break down that macromolecule into the individual nucleotides. So when we talk about a nucleotide, slide please, we talk about the three main parts of that. We have a phosphate. For DNA specifically, we have a deoxyribose sugar, hence the deoxyribo portion of DNA. And then we have a base, an A, a T, a G, or a C. Now, when we talk about the other nucleotides, we'll talk about a different type of sugar, but specifically for DNA, we have the deoxyribose sugar. So when you take these three parts together, Suter, and we talk about the phosphate, the deoxyribose sugar, and the base, we make a nucleotide, aka the monomer of the nucleic acid.
Nucleic acids is one of those
Macromolecules.
Oh yeah, I remember those.
Okay, so the different bases, A, T, G, and C, they stand for adenine, guanine, cytosine, and thymine. And I hope when you look at adenine, that looks familiar, that word anyway, and you're thinking of the other big A that we've talked about, which is adenosine triphosphate. And maybe a little later, we'll talk a little bit about the similarities between adenine and adenosine. But those are the 4 nitrogen bases: adenine, guanine, cytosine, and thymine. Slide, Suitor.
I was typing I say energy into the chat.
That's cool. Okay, so when we talk about DNA, there is something called a base pair rule. In other words, what are the nucleotides bases or the nucleic acid bases that are going to join with one another all the time? So adenine is going to pair with thymine in DNA, and guanine is going to pair with cytosine in DNA. The sides of this ladder, the sides that are connecting the DNA, are the phosphate and the sugar. And so if you look in the diagram to the on the right side of there, you're going to see like the dark blue and the light blue. That's representing the ladder. That's representing each of the sugar molecules where the base is directly connected to, and the phosphates are the lighter blue. And then the bases, you'll notice in the center, and they are labeled there with a key. So thymine kind of red, adenine's green, cytosine is yellow, and guanine is purple. And you'll notice that they are always paired with the same thing. Adenine is paired with thymine and guanine is paired with cytosine. And in between it, you have those little dots. Those dots are representing the hydrogen bonds that are connecting them together. Why hydrogen bonds? Well, when we talk about DNA replication, we'll talk about why they're hydrogen bonds and not a different type of bond. Slide.
So there was a good question here in the chat. Somebody said, do they have to be here if they've already done DNA structure notes and quiz? We did say that in the little thing we sent out yesterday. If we're done with DNA structure notes and the DNA structure quiz, yeah, we'll see you tomorrow.
Yep. Okay, and so when we're talking about this base pair rule, you should be able to look at the one side and figure out what the other side is. So if the one side is ATA, TCA, TGC, and GGG, what would the other side be? And I'm going to give a second to see if anybody can pop in on our chat and post what they think the other side would be based on the rules.
Like that, yes, in the chat. Instead of just randomly talking.
Random things. And those of you saying you're going to each other's houses, don't do that.
I have never started to randomly play guitar in class, and I'm not going to do that now. But maybe if you come to enough of these at some point, we'll play guitar for you. No promises.
Anybody got the other side? ATA, TCA, TGC, GGG.
Yeah, Broby, Juan Kenobi. You know how to type. Can you give us the other side of the DNA?
All right. Hunter, you started something and then you must have pressed enter.
The Supreme Ginger.
Yeah. ACG, CCC. Very good. No idea who Supreme Ginger is, but you're correct. Way to make assumptions. No, I'm just kidding. So that's absolutely correct. Okay, so A goes with T, so it would be TAT. G, thanks Ryan, you too. Okay, and we've got that all the way across: TAT, AGT, ACG, and CCC. And when we put those two together, oh, Connor, yes, mine.
Oh, come on, both were yours. All right, Kat, thank you very much.
And when we put those two together, okay, and they start to wind, then we get the shape, which I know was one of my daily questions last week, which is the shape of a double helix, and that is DNA.
Very nice. All right, so I guess I'm picking up here.
That's right.
Not that this is super important, all right, which is a great way to start out teaching you something, right? But as far as things that you would have to know if we're talking about the keystone and stuff, we're not sure this would ever pop up, but it might, and it actually is kind of helpful to understand. So there's two types of bases here, or nucleotides bases, the purines and the pyrimidines. And there's a really, really simple way to remember it, which makes it worth mentioning. So the purines, there's a little code here called PUGA2. And PUGA2, the PU stands for purines, the G stands for guanine, the A stands for adenine, and the 2 stands for 2 rings. So if you look at the pictures down at the bottom, you look at adenine and you look at guanine, and that little thing comes up all the time and gets in the way. You'll see that both of those bases have 2 rings. All right. So by default, then, the other 2 bases are going to be the pyrimidines with 1 ring. And so if we go to the next slide, one of the ways that you can remember which ones are the pyrimidines, they have a Y. That word has a Y in it, and both cytosine and thymine have a Y in it. So basically, we're just looking at ways to categorize this. And what's interesting, if you go back to what Mrs. Smith was talking about, and we talk about how base pairs match up and stuff, adenine always goes with thymine and guanine always goes with cytosine. Notice something. The pairs, there's always 2 rings in one and 1 ring in the other one. Right? So there are always going to be kind of like 3 rings in between each side of the DNA, I guess. Maybe that wasn't the best way to say it.
No, hold on, Mr. Studer. We've got Haley over there asking a question, and Astrid R kind of responded. I want to make sure Haley understands. So A, or adenine, always is going to bond with thymine. So if you go back and look at that, has like the example. So A is going to pair with T, so T goes there and T goes with A, and so you keep going on. And I see that you say it makes sense, and you got that from Astrid. So I just want to make sure that it doesn't matter. Whatever you have on the one side is going to be the opposite. So if it's A, it goes with T, T will go with A, C will go with G, and G will go with C. So
there's a really easy way to remember that, because A and T go together, and they spell the word at.
DNA is where it's at.
DNA is where it's at. And then C and G, they were sitting on the couch watching Netflix, they'd be spooning. So I know it's terrible. So last couple things here. In the notes that you're going to be filling out, it does talk about how many bonds we find between the different nucleotides. You can see that really easy on this picture. So between A and T, you have 2 hydrogen bonds. And between G and C, you have 3. So then the one question is, well, which one has a stronger bond? Right? And think about it a little bit. Would 2 bonds between be stronger, or would 3 bonds between be stronger? And that's pretty easy, right? I mean, that's one of those common sense things in science, right? That if it has 3 bonds, it's going to be stronger. The other thing that it talks about is this idea of anti-parallel. So I want you to look at this diagram and look at the oxygen specifically on the sugar. I think that's a great place to look. You'll notice that the oxygen is at the top of the sugar on this side of the DNA. But if we go to the other side of the DNA, the oxygen is on the bottom of the sugar. So one of these is facing up and one of them is facing down. And I think there's even a question that asks you, okay, so what's this 3' to 5' thing? Right, and so we see 3' at the bottom and 5' on the top on the left, and on the right you see it at the top and 5' on the bottom at the left on the right. And for right now, all I want you to take from that is it's directional. Right, it's just directional. We'll talk more tomorrow when we talk about replication and how DNA actually copies itself. That will be more important. But for right now, I think if you get that, it'll be good. Let me just take a look really quick at the notes and make sure that we hit everything that we had in the notes.
Yeah.
I think we did. I also want to just, because this is the first one of these that we're doing, and like we said, we're going to try and keep these reasonable. At 14 minutes, we're pretty good at that. How about just a quick shout out to people here? So I see Ryan Disney, I see Ryan Johnson, the Andres, Danielle, Hunter.
Kat was on here. Mark Baker's on here. Jessica's on here. Zuli's on here.
Wow, cool.
Hope's on here. Haley. I have no idea who Broby-Wan Kenobi is, but they're here.
Ucross TV. Ado. Zulika. So again, just to recap that, we're so happy you guys are joining us. We'd love for you to be here when we're actually doing them live. I think it's just normalcy to our day because I think our days are going to get kind of unnormal here in the future. So please come and hang out with us. Before we leave, I want to share one other thing with you. So let me change my screen around here.
As he's doing that, I know a couple of my honor students been asking about projects and everything. Yes, the due date's still Thursday. We're going to try and figure that out. I'm going to try and figure that out, and I will be sending you more information on that probably tomorrow. Give me a little bit more time to do that, because right now I'm teaching elementary school at home and bio with you guys.
All right. So real quick, if you go into Moodle, and I know Mrs. Smith's looks really similar to this, so hopefully it will carry over.
Yes, Elijah, we see you.
I got the office hours here. She'll send hers out. Once you get into week 1 DNA, you will see day 1 live stream Monday at 10:30. That's what we're doing right now. Here are the DNA notes. You have that posted too, right, Mrs. Smith? Filling that out. Really important. 2020-PD, your period number. If your period BC or DE, use the number I give you here, and your last name. Really important you label it that way, because if you don't do it exactly like that, we're not going to find it, and I'm not searching for you because it's just too hard to figure out who's participating and who's not. DNA structure slideshow from the live stream. That's the slideshow we were just showing you. And the last thing we would want you to do, so when you hang up or when we get off of this, if your notes are filled out, go take that DNA quiz real quick and see how you do. We want an 80% to move on. I think it's only like 10 questions. If you take it and you don't get an 80% and you review and you're still not at the 80%, that's when you would come into the office hours to talk to us and get some help. One other thing I forgot to do that I need to post in the chat is we did want to take attendance today. So in the chat room, I'm going to post a link. It's a Google form. It's our attendance form for today. Please go ahead and fill out that Google form before you do anything else, so we know that you were here. So we want to monitor that, because when we get back to school, we want to be able to say, look, these people did what they were supposed to do.
And I know some of you, at least for me, already have your DNA structure notes done and you wrote it down on paper. You know what I mean? If you're doing that, that's fine. Take it, your phones, take a picture of it, send it, you know what I mean? Label it and send it that way.
Right. You could upload it to Google Docs, give it the same title, and we'll make it really easy for us to find it.
Yep.
So any other questions? Johnny L's marking on my beard. It looks smaller in video. I trimmed about half of it off, Johnny L.
All right.
If there's no other questions, are we going to both be here tomorrow at 10:30? How are we doing that?
Yeah, I think 10:30 works. You know what I mean? And then we'll play it by ear from there and figure out what we're doing. But for tomorrow, we'll say 10:30 tomorrow. And then, you know what I mean? We'll kind of go through and see.
Tomorrow, 10:30, DNA replication. We're going to make this easy for you guys to follow. We're going to make it easy for you to understand. And we're going to keep doing what we do.
Thanks for being here.